In reality, I am ALS patient Guido Demetz. I heard my diagnosis begin 2000. However, this hasn't stopped me to obtain a PhD in bioscience. Currently, ALS means for me an electric wheelchair, help for almost anything and a weak voice. Therefore, I will be represented by my alter ego. After all, being an animated character has some advantages. Beside improving my appearance, age and health, I can move freely in the world of the imagination. And also very handy, I can speak clearly for a long time without weakening. The presentation, entitled ALS Research, The Way Forward, My Vision, will be explained by my alter ego. Although its contents is based on science, I emphasize that I am showing a personal interpretation. During this presentation, we discuss the following topics. In persons with ALS, motor nerve cells gradually die leading to progressive paralysis. Each year there are 3 to 8 ALS patients and 1.5 to 2 new cases per 100,000. Worldwide, there are approximately 250,000 existing ALS patients annually, among which roughly 50,000 new diagnoses. In addition, these numbers are increasing, with approximately 90%. Sporadic ALS surpasses largely the familial version, which is heritable. This has everything to do with mutations, hence a little bit of technical explanation. A human is a collection of 4,1013 cells, of which 1 in 40 nerve cells. Different kinds of cells exist, like muscle cells, skin cells and also nerve cells. These are supported by other cells, of which astrocytes and microglia are examples. Each cell is a protein factory. She needs them to function well. There are roughly 10 billion proteins in each cell at each moment. This means approximately 1,022 proteins in a human at each point in time. A protein is a chain of 100 to more than 1,000 amino acids. The making of a protein of 300 amino acids lasts approximately 30 seconds. Inactive and wrongly folded proteins are eliminated and its amino acids mostly recycled. DNA is a large molecule in the nucleus of each cell as part of a chromosome. As there are 23 pairs of chromosomes in a human cell, there are roughly 184 times 10 to the 13th DNA strands in a human. A gene is a part of DNA and is a code for proteins. There are approximately 20,000 human genes. A protein is only made when the corresponding gene is active. Gene expression, using mRNA as a go-between. The involved gene is transcribed on a messenger RNA molecule which is outside the nucleus, translated into a protein. Different kinds of cells have the same DNA but activate different genes. New cells are made before birth, during embryo development and during life using stem cells. For that, DNA is copied and copy errors, named mutations, are sporadically made. Motor nerve cells are not renewed during life and do not mutate. Therefore, a mutation in a motor neuron has to be present at birth. A mutation changes a gene and the corresponding protein encoded. Somatic mutations are not heritable, do not occur in germinal cells and have only an effect in mutated cells. Germinal mutations, on the other hand, are heritable and happen in germinal cells. ALS is probably a disease in 2 steps. First, motor neurons are attacked. This can have multiple causes and there are most likely differences between familial and sporadic ALS. Moreover, we deal here with ALS persons with no illness indications. Secondly, a disturbed communication between motor neurons and supporting cells results in faster dying of motor neurons. Motor neurons and supporting cells communicate with each other and they influence each other gene expression. When too much motor neurons disappear, their communication gets disturbed. The expression of some genes of motor neurons stops while others are activated. Motor neurons do no longer operate properly in the microglia. The immune system of the nervous system no longer recognize them and destroy them. The attack on motor neurons still continues, but its effect is now negligible compared to the destructive activity of the disturbed communication. There are still a lot of questions over when step 2 begins and gets the better of step 1. Patients in an early disease stage might still be in the transition phase. However, in my opinion, are all ALS patients with noticeable paralysis in step 2, and there are then no longer differences between familial and sporadic ALS. Familial ALS is caused by a germinal mutation in all cells which leads in the offspring to a change of the corresponding protein in motor nerve cells at birth. It is heritable. There is a familial ALS history and is in step 1 easier to determine for the known genes which are mentioned here. Sporadic ALS is probably caused by a local somatic mutation in stem cells which will form nerve cell supporting cells developed during life using stem cells. The activation of their corresponding proteins leads to cell death of motor nerve cells with which they communicate. Possible examples are mutations of genes of astrocytes which are responsible for the indirect connection between nerve cells and blood, alimentation, oxygen, waste products, carbon dioxide, and mutations of genes of microglia which vouch for the recognition of the own neurons. It is not heritable. There is no familial ALS history and is, like cancer, in step 1 difficult to determine. Currently, no gene mutations are known for this. The first generation of familial ALS is actually familial ALS disguised as sporadic ALS. A heritable germinal mutation, present at birth, causes a damaging change of the corresponding protein in all cells, thus also in nerve cells. There is however no familial ALS history, so it can only be determined in step 1 by screening. Is this a gray area of 10%? Also, casual multigene causes can currently not be excluded. Summarized, we have 4 ALS target test groups for clinical studies. Sporadic ALS step 1, sporadic ALS step 2, familial ALS step 1 and familial ALS step 2. Almost all studies use MSOD, 1 transgenic ALS models, from ALS step 1, and test only ALS patients with visible symptoms, ALS step 2. However, MSOD, 1 transgenic ALS models cover only 2% of the ALS cases, and the assumption that familial and sporadic ALS have an equal development is, according to me, also wrong. So, the answer for the question why do successful studies with mice produce no results for ALS patients is probably the following. Pre-clinical studies on MSOD. 1 ALS mice in step 1 are tested in clinical studies on familial and sporadic ALS patients with visible symptoms in step 2. So, it is no surprise that clinical studies have no results. Preventing and delaying ALS is only feasible for familial ALS step 1, including the first generation with no family history. In theory, it is also possible for sporadic ALS patients in step 1, but this stage is very difficult to determine. Possible approaches for preventing and delaying ALS are: deactivate mRNA of the active mutated ALS gene using antisense technology, augment the capacity to clean up inactive and wrongly folded proteins, and the long-time application of neuroprotective medication, among which certain, like tolampinol, beta-lactam antibiotics, coenzyme Q10 and minocycline, were vainly tested on step 2 ALS patients. Newly proposed treatments in this category are the upregulation of cell stress responses and mediction, among which lithium and rapamycin, promoting autophagy and mitochondriogenesis, This however requires the identification and recruitment of familial ALS Step 1 persons and the adaptation of the vision about clinical studies for the prevention and delay of ALS. The current clinical studies compare sick persons receiving placebo and medications and guarantee safety and effectivity for the ALS person. This means that you already have to be sick, which is mostly too late for the ALS persons, in order to be recruited in clinical trials. Only the persons having early markers, which are not known yet, can be tested with the protocol of today. In this regard accomplishes the research of Professor Van Damme, who tries to link certain harmful protein residues to a group of ALS patients, pioneering science. At one side, skin or epidermal cells are taken from persons in order to link the mutant C9orf72 gene, roughly 8% of the ALS population, to the accumulation of deviating protein remains in cells. At the other site, ALS patients recently diagnosed and with an C9orf72 mutation are recruited for a clinical trial investigating a C9orf72 antisense treatment. Proving that a treatment may lead to the prevention and delay of a disease is however a lot more difficult. Safety for the ALS person can be demonstrated, but showing its effectivity is harder. The main problem is the duration of the study. Visible ALS symptoms may occur very late or even never. As an extreme example, I mention a 10-year-old ALS person who gets the treatment during his lifetime, dies at the age of 90 and never has ALS symptoms. In this context, it will last 80 years before final conclusions can be drawn. Moreover, there is a problem with the recruitment for such clinical studies, as we probably find not enough familial ALS Step 1 persons who are not sick yet. In short, we need a protocol for clinical trials with treatments that might prevent and delay a disease. I plead to allow the administration of medication for ALS persons in Step 1 when its safety for the ALS person and its effectivity for the ALS animal model is proven. Is it not a pity to deny medication to ALS Step 1 persons? That may lead to the prevention and delay of ALS because of the lack of a suitable protocol. Perhaps a similar approach as for the approval of vaccines could be applied, in which only the expected effect is achieved before one is ill instead of recovery for one that is already ill. Some neuroprotective medication, already vainly tested on Step 2 patients, might indeed function for Step 1 persons. Patients with visible ALS symptoms cannot be cured and will not recover. The motor nerve cells and their connections that are lost will not return or be replaced. Only treatment can bring some comfort. For them, we have to take a different road and focus more on a better quality of life instead of a cure. Possible ideas for this are: suppressing the destructive activity of microglia, improving the communication between motor neurons and supporting cells, improving the recognition of the own motor neurons, and improving the quality of life by driving devices, such as exoskeletons, speech computers and breathing devices with a brain. Also, the own muscles should be considered. A possible name for this could be the neuromuscular performance enhancer. Most ALS cases are probably caused by certain local somatic mutations in genes of a stem cell during lifetime cell renewal. It is naive to think that somatic mutations only occur in cancer. Some mutations also might cause autoimmune diseases, like MS, and neurodegenerative illnesses like Alzheimer, ALS and Parkinson. All these diseases have in common that they are caused by damaging somatic mutations that slip through the net of the DNA control during the process of cell replacement. We most likely need an on very small nanobits based system in order to trace harmful somatic mutations in all relevant cells, destroy or repair them, report their action and disappear without leaving damage to fight such diseases efficiently. Therefore, Medici have the courage to leave the traditional policy of compartmentalization and cooperate with other specialists and engineers in the search for treatments. Also for ALS, this does not mean that the development of a cell scanner is simple. Dealing with a very large number of cells, intervene goal-oriented on a very small scale, and this both on an acceptable speed, will be required. This leads to the following conclusions. Expand ALS animal models to all ALS groups. Test the treatment on the proper ALS target test group. Consider all diagnosed ALS patients in step 2. Use the medication meant for ALS patients only when animal models with already visible symptoms are positively tested. Identify and recruit familial ALS step 1 persons. Involve them in the proper study and administer them medication which is safe for the ALS person and is effective for the ALS animal model. Adapt the protocol of clinical studies for the prevention and delay of the disease. Repeat the studies about neuroprotective medication with familial ALS step 1 persons and cooperate with other disciplines to find solutions. Learn as an ALS patient to live with a deteriorating health. Anticipate, use all available help, have regularly social contact, make sure that you have a daily, meaningful activity. Focus on what you still can, not on what you no longer can. Be tolerant and patient for yourself and your neighborhood and do not become depressive. Participate in clinical studies that provide a chance for success. This means studies that focus on suppressing microglia, improving the communication between motor neurons and supporting cells, improving the recognition of the own motor neurons and driving devices and the own muscles with the brain. Do not participate in clinical studies which aim at an effect on step 1 unless you have been recently diagnosed. You will only influence the result negatively and moreover, you might be disappointed with the negative result. Finally, hope that the neuromuscular performance enhancer will be developed soon to improve your independence and quality of life. An ALS cell scanner may be no longer useful for an ALS patient, but can, as long as a regular and effective screening is applied, eliminate ALS once and for all.